In this part 1 of the blood lecture, I expect you to be able to define blood, plasma, hematocrit, erythrocyte, leukocyte, platelet, thrombin, and hemostasis. Be able to list components of blood and define a function for each of those components. List the different blood cell types, red blood cells specifically, some white blood cell types, and platelet-forming cells. Be able to diagram how different blood cells are formed, kind of in general. Like erythroblasts make red blood cells, megakaryoplasts make platelets, and different white blood cells have a different source. And be able to list the steps in the process of clotting. So why do we have blood? Well, each cell in the body needs delivery of nutrients and oxygen. If you are fresh off the muscle lecture, you recognize that the delivery of oxygen and nutrients to slow twitch muscles is necessary for those slow twitch muscles to continue their long-term function. And likewise, each cell will need waste removal. For fast twitch muscles, if they're producing lactic acid, that lactic acid and other metabolic byproducts need to be removed. Blood acts as an intermediary between organs. If you remember back when we talked about hormones, the hormones are being delivered from endocrine glands to their target organs. Blood is also a defense system in the body, allowing the body to react systematically to a large-scale threat. And for your questions, there are about 1.2 to 1.5 gallons of blood in the average human, representing about 10% of the body weight. Your average human can lose 40% of their blood, and then they die. That's at 40%, they die. So losing 30% is very bad. Losing 40% is death. So what's in blood? Well, we have a term called hematocrit. Hematocrit is the blood volume that is occupied by cells. That includes both red blood cells and white blood cells. It's almost entirely red blood cells. There's also the plasma, which makes up most of the blood. It's the medium for transport, mostly water. And if you're thinking, that sounds like you have an extracellular matrix or fluid and cells floating in it, is this a connective tissue? It is. And in that extracellular matrix, the plasma, there are proteins. These proteins aid in clotting and aid in water balance because blood, one of its functions, is water balance. Let's look specifically at red blood cells. So there is a certain number of red blood cells, a certain percentage of the blood made up of RBCs, which we call hematocrit. That also includes white blood cells. These cells transport oxygen and therefore have a role in oxygen homeostasis. If you remember homeostasis, there is a set point, which is how available oxygen should be. A stimulus, let's say oxygen decreases in the blood. This is going to be sensed by a sensor, which is oxygen-sensitive cells in the kidney, and that will relay the message to a control center, which is also in the kidney, which will release erythropoietin. This stimulates more red blood cell production in the red bone marrow of long bones. And that will be a response, which would be the increased number of red blood cells, which will return oxygen availability to normal. What may cause this change in oxygen homeostasis? Well, perhaps moving to a higher elevation. At higher elevation, there's lower oxygen in the air, and since there is lower oxygen in the air, there is going to be, lower oxygen in the blood. This can actually mean you can move to a higher elevation and have more red blood cells produced, then go back to a lower elevation and have a higher ability to carry oxygen. This is called blood doping. Red blood cells themselves are also known as erythrocytes, and they primarily carry oxygen. They do also carry some carbon dioxide, but most of that will be in the plasma. Erythrocytes are small, they're flattened, and they're donut-shaped. They have no nucleus. They don't divide. What they do have is a lot of hemoglobin, and hemoglobin is a specialized protein that carries oxygen. No, not hemoglobin, hemoglobin. Made of 4 subunits. This has a quaternary structure, and 4 hemes, shown here as heme groups with iron atoms. Each one of these hemes is capable of binding 1 oxygen molecule, and this means one of these proteins binds a heme, which binds an oxygen molecule. These 4 hemes can bind 4 oxygen molecules at a time, and they bind to oxygen when the oxygen concentration is high and release oxygen when the oxygen concentration is low. So they would bind to oxygen in the lungs where they are in the capillaries next to the high oxygen air, and they release oxygen in the active body tissues, where those active body tissues are using the oxygen in oxidative phosphorylation to make water and to make more ATP. A specialized disorder called sickle cell anemia is a single nucleotide change in the DNA coding for hemoglobin. What this ends up doing is causing the entire erythrocyte, or red blood cell, to bend and to sickle up. So this single amino acid change that results from a single nucleotide change ends up making these sickled cells. If an individual has 2 copies of this sickle cell allele, they will have all sickled cells, which is problematic for breathing and moving oxygen around and can be lethal. If an individual has no copies of this, they have all normal red blood cells, which is great, except in areas with malaria. In areas with malaria, the malaria parasite actually cannot parasitize sickled cells. So if one has 2 copies of sickle cells, that's bad for them. If they have 2 copies of normal cells, that's going to make them more likely to get malaria. If they have 1 copy of each allele, they do get slight sickling, but not enough to be lethal. And they can get malaria, but not as badly as some other individuals. So it actually gives an advantage to heterozygotes. We will cover heterozygosis later on in another lecture. Right now, let's look at the other cells. So we see stem cells in the red bone marrow. These stem cells can form erythroblasts through differentiation. They just turn into different cells when they divide. Those will lose their nucleus, fill up on hemoglobin, and be erythrocytes, red blood cells. Stem cells can also form, there at the bottom, megakaryoplasts, which will turn into megakaryocytes and give rise to platelets. There are also several different ways to become a white blood cell. And you do not need to memorize this. You just need to know that it's not the same way as becoming a red blood cell. You have myeloblasts, giving rise to granular leukocytes, monoblasts and lymphoblasts, giving rise to agranular leukocytes. These are all white blood cells, or, as they're also known, leukocytes. So leukocytes are the white blood cells in general. They make up 1% of whole blood. Neutrophils engulf foreign bodies by phagocytosis. You see a picture of a neutrophil here. You may hear them called macrophages, or big eaters. Eosinophils defend against large parasites and moderate allergies. Basophils mediate histamine release, one of those hormones that helps cause immune responses. Monocytes go out of the bloodstream and into the intracellular spaces and engulf invaders. Lymphocytes deal with bacteria and viruses, or viriae, as B cells and T cells. Looking specifically at these B cells and T cells, B cells will recognize antigens, or foreign things that need destruction, in the bloodstream. In response, they will produce antibodies that bind to those antigens. This is negative for the antigens because they get bound up and eventually eaten by macrophages. Some B cells will persist after an infection and will serve as memory, thus causing a faster response if the infection occurs again. T cells recognize antigens presented by cells. A cell that is invaded by viruses will actually display parts of the virus on the outside of that cell. The T cells will recognize, hey, this cell is infected by a virus, and they kill the cell, which kills the virus within. So this does destroy the cell. And after an infection, some T cells will persist and serve as memory, which will enhance the response if viruses affect the body again. A disorder of white blood cells is leukemia. Acute leukemia is a very rapid production of white blood cells that causes, although the hematocrit may be the same, the proportion of white to red blood cells is favoring white blood cells and not favoring, well, oxygen transport. And this can kill within a few weeks. Chronic leukemia takes 3 to 10 years to develop. The chemotherapy is one option, including leucocristine, which comes from this Madagascar periwinkle. There is also bone marrow transplants as an option. You should register on the bone marrow registry. Look into that because you may be able to save a life. Let's talk about clotting. Hemostasis, or blood clotting, is the stopping of the flow of blood, and this happens through several steps. First off, there is a vascular spasm. Smooth muscle that surrounds the arteries or arterioles will constrict, thus reducing the blood flow to the region where there is a cut. Then a plug of platelets will form. This plug of platelets will stop cells from leaving, and then a clot will form with fibrin, causing an insoluble mesh and trapping red blood cells and platelets. Let's look more specifically. Here is a clot. We remember that platelets are cell fragments formed by megakaryoplasts that will fragment into small chunks of cells that will float in the plasma as platelets. Thrombin is an ever-present enzyme that is inactive except when cuts are made into the capillaries. This will turn fibrinogen, another protein that's constantly present, into fibrin, which is an insoluble mesh, and will make a net that will trap red blood cells, trap more platelets, and fill the gap with a bunch of trapped cells, kind of like an ultra traffic jam. Aspirin can actually reduce the action of thrombin, and it is recommended that you do not take aspirin if you are giving blood, or you do not take aspirin if you suffer from frequent bleeding. Aspirin slows blood clotting and can actually be used as a medicine to prevent strokes and heart attacks, which can result from blood clots. Tea also contains something called tannins, which give it that astringent flavor. Teas that are not astringent, like, say, green tea, will not have as much tannins and therefore will not be effective to bind proteins. That's what tannins do. They actually bind proteins together, which is why a tea bag on a cut can stop bleeding faster than a bandage on a cut. Tea bags contain these astringent chemicals and are actually also antibacterial. So if you need some good first aid and you happen to have some tea, applying a tea bag to a cut can aid in the clot and be an antibacterial.